Jones from the University of Massachusetts Medical School. And Dr. Jones and I have known each other for about 10 years. It's an honor to present Dr. Jones to you. He'll be speaking on genetically similar, epigenetically different insights into FSHD from studying discordant families. Thank you.
Okay, well, first off, I'd like to thank you for the invitation to come speak. And I always like to start out by pointing out that we function in my lab as a husband and wife team, co-PIs. My wife, Takako, is back there in the corner. And we pretty much do everything together, and it works out. So I don't know if that's unusual now, but it's we have a good time. And our main, we do, seriously. And so our main interest is actually on epigenetics of FSHD. And Silver talked a little bit, he kind of touched on epigenetics as well as genetics of FSHD. And I would argue that epigenetics is at least equally as important, if not more important, actually, in respect to FSHD. And we consider FSHD actually an epigenetic disease. So epigenetics has been around about as long as genetics. It's this idea of non-Mendelian inheritance. And it's really, though, only in about the past 15 years that there's been this real explosion in epigenetic research, in biomedical research, and also in pharmaceutical companies developing small molecules that can target epigenetic processes. And so what I thought I'd do today, since you guys probably have run across epigenetics quite a lot just in the everyday news, as shown here, and you're going to run across it all the time if you're paying attention to the FSHD literature, I thought what I would do is basically make sure you all understand what we're talking about when we say epigenetics. Okay. So normally the way I would do this is you would take my 40-lecture full semester course, which you're welcome to do. And in that course, we'll do yeast and worms and flies and stuff, and we'll use all these as models for how we study epigenetics. But since I only have about 10 or 12 minutes here, I'm going to go with the simplest model that we use, and that's Paris. And the thing about it, she catches a lot of flack, I know, but if you really look, she's actually pretty complicated, okay? You're looking at about 10 to the 13th cells, making up Paris. And each one of those cells, and there's hundreds of types of cells, and each one has 10 to the 12th proteins. And you got to think the different cells have a different set of 10 to the 12th proteins, right? You only have around 22,000, or God knows how many you actually have now. Depending on who's counting, you got around 22,000 protein coding genes. Almost all of them make more than one type of a gene product. That's alternative splicing. You have these, now we know there's about 40,000 non-coding RNA, so most of your genome doesn't even encode a protein. You have, 500,000 regulatory elements, and this is essentially one genome. And through all of this, you still have to basically, every cell has to make the necessary genes that make that cell function. And one of the things that I think a lot of people kind of ignore, forget, but again, Silver brought up, is that it's not just making the genes that a cell needs, you have to actively keep off the genes that a cell doesn't want, this repression. And that's actually essentially what FSHD is, this failure in this repression. And so your cell actually has this mechanism to do that, and that's essentially the epigenome. Okay, so the epigenome makes all of this work. Essentially, it organizes the nucleus. It will affect the mRNA content, but really the keys of the epigenome are that it's a mechanism that your genome and the different cells can actually integrate signals from the environment. And what really makes it epigenetic is that this provides a mechanism for cellular memory. Cells, after division and throughout life, can remember events that happened many, many divisions earlier, even in utero. And also, some of these events can be passed on to the next generation of actually your kids. And so what this is that events that happen at points during cellular development can be remembered. And in the case of FSHD, what a cell might want to remember is to keep DUX4 off. and epigenetically keep this off. So basically what epigenetic gene and genome regulation is, it's the structural adaptation of chromosomal regions so as to register, signal, or perpetuate altered activity states, as defined by Adrian Bird. So the keys, now your genome is your DNA sequence. The epigenome is DNA sequence independent. Okay, it is context dependent. So Ver showed you that spring. Where you are in that spring matters if you're a gene on whether you're going to be expressed or not expressed. Okay, these are stable and heritable among cellular generations. Some of these events are stable to multiple to your offspring, not just cellular generations, but onto the next generation of the person. But they're stable and heritable, but they're also dynamic and reversible, which sometimes doesn't make sense. Right? These are very stable, but you can flip the switch. And if you can flip the switch, then you can actually inherit or stably perpetuate an altered epigenetic state. And the other key to these are that they are responsive. These signals are responsive to environmental stimulus like sleep, stress, diet. So your epigenome, your DNA, is actually responsive to the environment. Okay, so the best way that I know to show this is from Randy Heritable. I love this picture. These are actually genetically identical mice. Okay, they are epigenetically different. And you can do this in mice, and hopefully not in people, but we do this in mice. You can actually look at this guy here that's a little yellow, the mottled one, and the brown one. These are genetically identical at the sequence level. They're epigenetically different. Okay, we call these epialleles. And the thing about the epigenetics is that this has profound long-term health consequences, because it's not just that this guy on the left here is going to be yellow. He actually is going to spontaneously develop tumors, okay? And he's going to be obese. And his brothers that are actually they're not brothers, but they're genetically identical mice that are epigenetically different are not going to be predisposed to tumors or obesity. And the way that these were different is they were different in what mom had or what mom was fed while they were in utero. So the environment of them during development dictated, I don't have a pointer, but dictated whether or not you had an epigenetic mark as signaled by these stars or not in the lowercase. And whether or not you had this epigenetic mark was determined on mom's diet, and then the outcome is during development, spontaneous tumors or not, obesity or not. This is epigenetic, okay? Genetics affected by mutagens and mistakes, that's your nature. Your nurture affected by stress, sleep, diet, prenatal care, environment. They are both able to be passed on by mom and dad. The difference is the epigenetics can be switched. Genetics, you got to do gene therapy. That's a tough thing. Epigenetic, but I even say that with FSHD or anyone with an epigenetic disease. You're lucky if it's epigenetic because you can switch it, you can flip it. Remember, these are dynamic, these are reversible things. So if we can figure it out, it should be reversible. There are a lot of epigenetic diseases. I could have probably 10 slides now. This is my old slide. FSHD is the one we work on. But there's a number. You'll see down at the bottom. There's obesity, diabetes, bipolar. Basically, epigenetics is involved in human health and disease. Okay, and this is actually good news. And I'll explain why in a sec. Okay, so Sylvain had mentioned again the genetics of FSHD. So FSHD1 and FSHD2, while they're genetically different, they're actually linked by epigenetic dysregulation of the 4q35 locus and DUX4 gene expression. Okay, and so in a healthy, unaffected individual, essentially you're epigenetically repressive. This region was programmed to be off during development, and it remembers that it wants to be off throughout life. In FSHD1 and 2 affected, this program has been disrupted, and this region is now epigenetically amenable to being active. Doesn't mean it's necessarily active, but it's amenable to being active. It's open to it. So what we were interested in is looking at these people that are asymptomatic. Turns out there's quite a number of people in the population that have the genetic requirements for FSHD1, but do not ever develop disease. And so one of the questions is, how do these, since we know that epigenetics seems to be what dictates whether or not you develop disease, let's look at the epigenetics of these unaffected subjects, or asymptomatic subjects. So what we're going to look at, again, these are FSHD1. Genetically, you would be diagnosed as FSHD1, but never develop any clinical manifestation of the disease. Okay, and it's actually quite possible that a sibling could have the same genetic mutation and have the disease and actually have be quite affected. This could also be through generations where you see differences in being affected, that the same lesion tracking through multiple generations doesn't necessarily give the same disease presentation. So we're curious, could this be epigenetic? And so what we're able to do is we're part of the Wellstone Center that you heard about at the very beginning at UMass Medical School. We have this large collection of families that have donated biopsies and blood. And so we have first-degree relatives. And you'll see one of the things you see by this is you'll see there are a number of labs doing a number of things working together, and that's essentially what a Wellstone Center is. And we're down here in the corner doing the epigenetic analysis of these things. And so we're able to look at first-degree relatives that have the same genetic mutation, so they're genetically FSHD1, but clinically either have no presentation of the disease, asymptomatic, or actually quite symptomatic. And all you need to do when you're looking at this is say, what we do is we track down the DUX4 gene, the pathogenic DUX4 gene, and we do a base-by-base readout of the epigenetic marks. And if the epigenetic mark says this should be off, this is repressive, this should be silent, you'll see it as red. And if the epigenetic mark says this should be open, this should be on, then you'll see it as blue. And all you have to see right away is in healthy controls, it's mostly red. The DUX4 gene is mostly epigenetically off. And in subjects that are affected with FSHD1, they're mostly on. So this is a clear epigenetic difference at the pathogenic DUX4 gene that shows the difference, and this correlates with expression, the DUX4 gene being on versus off. Then we looked at asymptomatic subjects and families, again, first-degree relatives, and we've actually looked at now about 14 of these families. And we see pretty much exactly the same thing. What you see is on the left, FSHD-affected, looks just like the FSHD-affected previously. Very epigenetically open, amenable to expression of DUX4. But what we see is just a little bit more red on the right. And if you look at the numbers, what you see is essentially it's twice as much. Now, if we looked at the numbers on the healthy controls, you're about 70%, very, very off. We look at our asymptomatic, it's actually not that much different. It actually says epigenetically, you only need a little bit of a nudge, or a little bit of a nudge either way, and that's the difference between whether you develop clinical FSHD or not. And this is actually really good news. We actually are able to group this up. This is not coming out quite as I had planned, but essentially if we have a significantly distinct grouping of FSHD affected versus healthy, and the asymptomatic is actually very close to affected. So what this says is you don't have to epigenetically change someone that's affected very much to actually flip the epigenetics back to a point where they might no longer develop symptoms. Okay, so basically this is what we think of this looks at. Our healthy is very epigenetically off. FSHD1 and FSHD2 is very epigenetically on and open and expressing DUX4, and the asymptomatic FSHD is an intermediate stage. Okay. So does this have implications for therapeutic development? And absolutely it does. Okay, what I had told you, remember, these are epigenetic diseases, good news. Okay, these are dynamic, reversible. That means these are druggable targets. The epigenome is druggable. Okay. So that's good. So this is, I was thinking about all the different ways that you could target FSHD or, pathways that you could affect for FSHD. These are kind of what I came up with as my cartoon, and there's a lot. And we don't know which one's going to work, and we don't know which one is the best one, and which one. You're going to hear a lot today about targeting DUX4 expression and targeting whether it's mRNA or the protein or protein function. What I'm going to say is that actually targeting the epigenetics is actually a viable approach. This is actually all the way upstream, because if you can fix the epigenetics, you can target the epigenetics, everything downstream comes with. Okay, so we're going to I'm going to make the case that actually epigenetic regulation is a viable therapeutic target for FSHD. And we're not just blowing smoke when we say this, okay? I mentioned in the past 15 years that there's been an explosion in epigenetic research and looking for drugs. There are a lot of epigenetic diseases. This is the number of clinical trials that were going on in 2010. There were over 500 clinical trials using epigenetic drugs to treat epigenetic diseases. This is where pharmacy is going. Okay, and if you look down here where Yang Shi, he's an expert in, a leader in personalized epigenetic drugs. And it's his quote, actually, I love his quote. He says, every big pharmaceutical company has a robust program in epigenetics. It's quite a change in the past few years. Epigenetics and epigenetic targets, epigenetic drugs. So what this means is all these other diseases, all this other research, all these other molecules, some of these may be applicable to FSHD. Okay, we can actually, you're not starting from scratch. You're able to feed off these other research. Okay, so in summary, epigenetic regulation is context-dependent, sequence-independent, stable, heritable, yet reversible. That's question 1 on the exam. Define epigenetics, that's your answer. Okay, and actually someone will miss it, I know, but that's okay. I've had a lot of kids. FSHD is an epigenetic disease. The overall epigenetic status of the D4Z4 correlates with clinical presentation of FSHD. Small but significant epigenetic differences between being asymptomatic and FSHD-affected. The status of the D4Z4 is truly a viable therapeutic target, and that many drugs targeting epigenetic modifications are already being developed for many other diseases. Some are in clinical trials, some are FDA approved. And if we can find the right one, we jump ahead. Okay, so this is good. So I'd like to point out that, so this work, as was Silver, is going to be working on getting it published soon. This is the group at the Wellstone that have been working together on this. I want to mention the people in red are all here in this room. So if you have questions about epigenetics, feel free to ask any of them. There's a lot going on with epigenetics, and this has been a pretty good group to work with. And this is my little group, in our lab selfie. And we'd like to acknowledge our funding from NIH, particularly the FSH Society, the Chris Carino Foundation, AFM, and MDA for funding our epigenetic work. And thank you very much.